Hello friends, welcome to my YouTube channel, and today we are going to learn amino acid structures and properties very quickly. Yes, the key idea of this lecture is to quickly learn structures and properties of amino acid. So let's begin. Before we go to the structure, we just take this idea. So there are polar molecules and non-polar molecules. Mostly these are hydrophilic, they are more soluble in water, and they have an OH group, or they have charge, either positive or negative charge, but they are charged molecules. Whereas non-polar are mostly hydrophobic, hate water, they are less soluble in water, and they have aliphatic chain. Longer this chain, more hydrophobic this molecule will be. Now let's see, if we see amino acid, there is a carboxyl group, there is an amino group, a hydrogen atom, and an R group. If we see the amino acid structure, this part, the carboxyl group, amino group, and hydrogen atom is common to all the amino acids. So basically, if we see, an amino acid is negatively charged as well as positively charged. It's an amphipathic molecule which contains both the charges, positive as well as negative. But the properties of amino acid is mostly decided by R group, because this is the only group which is going to be varying. So the first structure which I want to show is glycine. A glycine is one of the amino acid which has an H instead of R. So this is the only amino acid which is not asymmetric, which means all other amino acid have 4 different groups attached to the asymmetric carbon atom except glycine, right? Since this is a very small molecule attached to it, this is the smallest amino acid also. If we check alanine, this has a CH3 group instead of H. If you see, most of the amino acid have this basic structure. Now, if I ask you whether glycine or alanine, which one is more hydrophobic? Yes, it would be an alanine. Why? Because it has a aliphatic group attached to it. Now let's see the next structure. As I said, that the most of the amino acid have this sort of structure which is common to all of them. Now this one is valine. Valine is an amino acid which you can remember by making an alanine, and which has a V. V for valine, and this V is basically 2 CH3 group attached to it. Now again, if I ask that which one is more hydrophobic, alanine or valine, so we can understand that now this has 2 more groups attached to the alanine, which are aliphatic groups. So this will be more hydrophobic. The next structure is leucine. You can see it. You can find the similarity with valine. Yes, This is same as valine except this has an extra CH2. So if we add one more aliphatic group CH2, then this become leucine, right? Again my question would be same. Which one is more hydrophobic, leucine or valine, right? And you know the answer. The answer is leucine will be more hydrophobic because it has one extra methyl group. Let's see the another structure. The other structure is isoleucine. So remember isoleucine, remember that this one has an L group. So if you make an L, this would be a structure of isoleucine in which we have everything. If you see the molecular weight, molecular mass, it is same as leucine. It is same as leucine, right? Same number of carbon atoms. So if you see the R chain, there are 4 carbon atoms like leucine, and same we have in isoleucine, right? And this, if we see that these molecules are isomers, their molecular weight is same, their most of physical properties are same, also the hydrophobicity is also almost same. The only difference is a leucine which has an iso group, which is a branching of the methyl group. Another structure is methionine. Now methionine is one of the structure which contains sulfur, right? This contains a sulfur. So this is one of the sulfur containing amino acid. But this sulfur is occupied by a methyl group, unlike another sulfur containing amino acid which is cysteine. Now if you see cysteine, this also has an S, but this S is free because it is not occupied by a methyl group. So this can get a charge when once it lose an H plus, what would be the charge? It will become negative, right? Remember about the solubility. If there are charge, it will be more soluble. So if I ask you in comparison to methionine, cysteine would be more soluble. Another reason is that it has less number of methyl group. So this is charged, as well as methionine has more number of CH2 group in comparison to cysteine. So methionine will be more or less hydrophobic. Yes, methionine would be more hydrophobic than cysteine. Another property is that this cysteine can make disulphide bonds. So proteins, disulphide bonds are made up of cysteine. Why not methionine? Because methionine has a methyl group, right? There is a methyl group here which is which prevents it in making a disulphide bond. So if we compare methionine and cysteine, which one is more hydrophobic? Methionine. Now let's think from an enzyme perspective. So the enzyme, an enzyme which has an exposed SH, right, like cysteine, would have a better chance to make bonding with the substrate for charge transfer, and so most of the enzyme will have cysteine in their catalytic pocket and not methionine. Methionine will only be in the catalytic pocket of enzyme where hydrophobicity is required. But in general, we know, which we are going to discuss in our enzyme lectures, that one of the way in which enzyme catalyze any reaction is by lowering down the activation energy, right? and these charges actually help in progress of that reaction. At present, you don't have to bother about that, but the understanding that how these molecules are very effective in the enzymatic pocket, cysteine. Now, another similar structure is serine. So, if you see serine and cysteine, there is only one difference. That is, instead of SH in cysteine, serine has an OH, right? It is an alcohol containing amino acid. Now, remember again about the solubility. If there is an OH, these amino acid will be more soluble, right? Polar group. So OH is a polar group. So this is also a polar amino acid which has an OH and a better solubility. Another alcohol containing amino acid is threonine. To remember the structure of threonine, it's very simple. It's simple that you make a T like this. Okay, T for threonine and T for T is the structure. So one side you make an OH and another side you make a methyl group, and this is the structure of threonine. Okay, so this is the structure of threonine, in which what we have is a methyl group and a hydroxyl group on both the sides. Again, if I ask you that which one has better solubility, serine or threonine, what would be your answer? Yes, both of them contain OH, but threonine has a longer aliphatic chain, which means it has more methyl groups, which means it is relatively hydrophobic, right? Now, we are not comparing threonine with valine or leucine or isoleucine. Of course, those molecules would be more hydrophobic. We are just comparing threonine and serine, which are both of them are hydrophilic molecule. But if we compare between serine and threonine, threonine would be more hydrophobic, serine will be more hydrophilic. Now, this structure is really interesting. So, this is aspartic acid, right? Now, as if you have observed that most of the amino acid which we have seen, you will find that a structure of alanine is common to most of them, right? So, if we add a carboxyl group COOH to the alanine, it becomes aspartic acid. Whereas if we add an amide group CONH2, this becomes asparagine, right? Now a similar structure to this is glutamic acid and glutamine. It's the same thing. This glutamic acid contains carboxylic group, whereas glutamine contains an amide group, right? Now here if you see what is difference between glutamic acid and aspartic acid, there is one extra methyl group, right? Same, what is difference between asparagine and glutamine? One extra methyl group. Now, if you see glutamic acid, so because of resonance, this can get a negative charge. This is a charge atom. So, in comparison to this, where this double bond will not make a resonating structure, which means what? That this will be charged and this will not be charged molecule. So if I ask you which one will be more hydrophilic, of course it would be glutamic acid. More polar, charged, and so it is more hydrophilic in comparison to glutamine, right? The but the difference would be more if you see glutamine and aspartic acid. So now aspartic acid has a carboxyl group, right? So it is charged, a polar group, in comparison to glutamine, which is uncharged, more number of um methyl group, and that's what make glutamine hydrophobic in comparison to aspartic acid, right? Now, as I said, that there are most of these amino acids have, you can again see this example has an alanine group in it. If you see, this is alanine, and if we add a phenyl to alanine, this becomes phenylalanine, right? Phenylalanine. Because of this aromatic large ring, this become hydrophobic. But if we add an OH, this become tyrosine, right? OH is a hydroxyl group, right? Which will increase the solubility. So if I ask you which one is more hydrophilic, tyrosine or phenylalanine, the answer would be yes, tyrosine is more hydrophilic. Why? Because it has an OH group. This is a very interesting structure, tryptophan. So tryptophan is a molecule, is an amino acid which is represented by a W. So you know that most of these um, amino acid, all of these amino acid are also represented by a single letter. Single letter for tryptophan is W, right? And if we make a pentagon at one side and a hexagon to other side, this become a tryptophan. This is a more defined structure, and also it contains an N in the pentagon. This is the structure of tryptophan, right? And alanine, remember this is an alanine which has a which is made up of a W. One side is a hexagon, Benzene, and a pentagon. This pentagon contains a M. If you see that this is the larger ring and which will have a better resonance in comparison to both tyrosine or phenylalanine, right? And because of this high resonance, we measure absorbance at 280 nanometer, we get an absorbance curve. So if you see this graph, this sort of graph would be for phenylalanine. Little better for tyrosine. But for tryptophan, it is huge. Which means most of the absorbance of any peptide which we measure is because of tryptophan, right? So even if there are two peptides, so consider there are two peptides like peptide 1 and peptide 2. This one contains more number of tryptophan and this one contains less number of tryptophan. Which one you think will have a higher absorbance at 280 nanometer. Yes, peptide 1. This is another molecule which is lysine. Lysine is an amino containing amino acid which has 4 methyl group. So this has a charge, positive charge, right? A positive charge amino acid. Another positive charge amino acid is arginine. Arginine has a 3-point star which has a carbon in between and a symbol like Mercedes Benz. So it has all the point of this star has an N. Valency of nitrogen is 3, remember, right? So accordingly we can uh, put up the hydrogen atoms, and if there are 2 hydrogen atoms there would be a double bond and a positive charge. This is another positively charged amino acid. There were 2 negative charge amino acid, remember, glutamic acid and aspartic acid, right? There is another one which is positively charged, and this is called as histidine. Now histidine is a pentagon, so what we have is alanine, right? An alanine with a pentagon, and it has 2 N group here. Histidine has a better buffering capacity, and it buffers in many biological systems. We will see this when we see pH and pK or the chemical properties of the amino acid. At present it is, and this is positively charged. So we have 3 amino acids which are positively charged, and the other one is 2 other 2 which is aspartic acid and glutamic acid which are negatively charged. Another interesting structure is proline. Now what proline does is it has a amine group and a aliphatic group. This aliphatic group comes and join the amine group. This is the structure of proline. So let's quickly see all these non-polar aliphatic group. It is glycine, alanine, proline, valine, leucine, isoleucine, and methionine. The common thing to them is that there are aliphatic chains. Now, if I ask you that alanine is more hydrophobic or isoleucine, the answer would be yes, isoleucine. Why? Because it has a larger aliphatic chain. Remember, there is similarly there are polar groups. Polar groups because of OH, right? Serine and threonine, cysteine, asparagine, glutamine, the amides, and we have negatively charged aspartate and glutamate. Aspartate which contains a carboxyl group, glutamate which contains carboxyl group. If I ask you a peptide which has more number of aspartic plus glutamate, this peptide will be negatively charged, anionic peptide, right? But if there is a peptide which contains more number of lysine or arginine, this would be a positively charged peptide, or what we call it as cationic peptide. Similarly, these are the three which have aromatic arginine, which we have already seen the structure: tryptophan, tyrosine, and phenylalanine. In which one has higher absorbance at 280 nanometer? Remember, tryptophan. Why? Because it has a huge aromatic chain for resonance, and because of that there is a high absorbance at 280. I hope you all enjoyed the video. Thank you.